In the pharmaceutical industry, especially when manufacturing sterile products, the environment in which these products are made is just as critical as the ingredients themselves. Maintaining a controlled and contamination-free environment is paramount for patient safety, and that's where environmental monitoring comes into play. Today, we're going to explore what environmental monitoring is and why it's such a vital aspect of pharmaceutical GMP. So, what exactly is environmental monitoring? In simple terms, it's a planned program of collecting and analyzing data about the environment within a pharmaceutical manufacturing facility. The primary goal is to detect and control contamination, both microbial and particulate, ensuring that the manufacturing areas maintain the required levels of cleanliness. This helps in verifying the effectiveness of cleaning and disinfection procedures, as well as confirming the integrity of the cleanroom environment. Ultimately, environmental monitoring plays a crucial role in supporting product sterility and overall patient safety, and it's a fundamental regulatory requirement across the globe. There are several key parameters that are typically monitored in a pharmaceutical environment. Viable monitoring focuses on detecting living microorganisms. This includes air sampling, which can be done actively by drawing a known volume of air over a growth medium or passively by exposing plates to the air for a set period. Surface sampling is another critical aspect, often performed using contact plates pressed onto surfaces or swabs that are then streaked onto growth media. Personnel monitoring is also essential, typically involving taking glove prints of personnel after critical manipulations and sampling their gowns. In some cases, water sampling might also be required, especially if water comes into contact with the product or critical equipment. Alongside viable monitoring, non-viable monitoring is also important. This primarily involves measuring total particle counts in the air to assess the overall cleanliness level. Additionally, parameters like temperature and humidity are often monitored as they can impact microbial growth and the stability of certain products. Pressure differentials between different cleanroom zones are also tracked to ensure the intended airflow and prevent the ingress of contaminants. Depending on the specific manufacturing process, other parameters might also be monitored. To effectively monitor the environment, it's crucial to establish appropriate sampling methods and locations. These are typically defined based on a thorough risk assessment, identifying critical areas where contamination is most likely to occur or could have the most significant impact. The frequency of monitoring is also determined based on the risk level and regulatory guidelines. Various types of sampling equipment and techniques are used, each with its own specific procedures. For viable monitoring, proper handling and incubation of samples are essential for accurate results. Despite all precautions, sometimes excursions or results outside the established limits can occur. When this happens, a thorough investigation of the root cause is necessary. This might involve reviewing procedures, environmental conditions, and personnel activities. Based on the investigation, corrective and preventative actions, CAPA, are implemented to address the issue and prevent future occurrences. It's also important to conduct an impact assessment to determine if the excursion could have potentially affected the quality of any product manufactured during that time. Finally, the data collected from environmental monitoring is often trended over time to identify any potential patterns or developing issues that might require proactive intervention. In conclusion, environmental monitoring is an indispensable element of pharmaceutical GMP. By systematically monitoring the manufacturing environment, pharmaceutical companies can effectively detect and control contamination, ensuring the sterility and quality of their products and, most importantly, safeguarding the health and well-being of patients.